Okay, welcome back to our lecture series DNA and Design, and we are now in the second lecture of the subsection titled Code Within a Code, Multi-Layered Information. So let us now begin exploring some of the details. We have said that DNA, while it carries the code for protein synthesis for the order of amino acids to make a protein, simultaneously also has embedded within it codes for multiple other functions, and we saw some of the jargon in prior papers. Let's now try to make sense of some of that jargon. And so the first function we want to explore is regulation of chromosome structure. When we looked at DNA, we basically looked at this idealized double-stranded structure with a whole bunch of nucleotide bases and looked at them, as we said, in a linear fashion. But we have to realize that the physical structure of DNA is very, very complex. If you were to take the nucleus of a cell, a human cell, and stretch out the DNA, it stretches to about 2 meters, maybe a bit over that. And all of that is very tightly coiled and packed into the nucleus, whose size is less than 1/10 the width of a human hair. And so, how do you take this long string and coil it up so tightly and so compactly, but in a smooth and organized way so that it is unfurled in different sections at will, so that those sections can be transcribed and translated when the cell needs the proteins? That is extraordinarily difficult. As I mentioned once before, anybody who has worked with a telephone that has a cord, or fishing line, or a garden hose, you realize firsthand that even with something non-intricate, with sizes much less than what we're talking about, these things get knotted up so easily. Every time I want to plug in my headset to tape these lectures, I have to untangle the headset cord. So the way that DNA does this is it winds itself around these protein structures called nucleosomes, and then has these nucleosomes pack in together to form chromosomes. So I know some of you have a mental picture of chromosomes. Well, those are the tightly wound structures of DNA. Now, this process doesn't happen haphazardly or at random. There's a great deal of information that tells the nucleosomes where to locate on the DNA strand, how much of the DNA is wound along each nucleosome, and how those nucleosomes get packed together to allow the DNA to be unraveled as needed and then rewound again without getting knotted and tangled. Now, where does the information for this organization come from? Well, of course, it comes from the DNA itself. DNA is the storehouse of information for everything that goes on in the cell. And so, in addition to carrying the nucleotide base sequence to make the proteins, simultaneously the DNA within that same sequence is also carrying the instructions for how it should be wound along the nucleosomes. Okay, let's now move to another code within a code. And remember, again, these are not things being coded by different sections of the DNA. This is the same code that simultaneously is conveying multiple different pieces of information, multiple layers. So the DNA code also regulates the speed of protein synthesis. This is a very important issue because as proteins fold, we've already talked about how they need to fold in the right way. Stable protein folds. And so as they come off of the ribosomes, as the amino acids are being linked together, the protein is folded piecemeal. and being able to accurately fold it into its very intricate secondary and tertiary structures that we've covered in a previous lecture requires that it needs to come off of the ribosomes at a given speed, and that speed varies with the section of protein being coded and the sort of fold being made. So how do we vary that speed of the string of beads of amino acids coming off of the ribosome so that they can be correctly folded? Well, it turns out that this is embedded in the redundancy of the genetic code. Now, before we go further with this topic, I just want to remind you what we mean by the redundancy of the genetic code. So if you look again at the table of 3-letter codons using the RNA alphabet, which is similar to the DNA alphabet except that thiamine has been substituted by uracil. So if you notice, for example, the amino acid leucine, you see that 6 different codons code for leucine. UUA, uracil adenine, codes for leucine, but also CUG codes for leucine, and so forth. So that is the redundancy that we talked about, that multiple different codons code for the same amino acid, so that whichever one of those codons is on the mRNA sequence would still bring that same amino acid leucine, for example. Now, we had talked about one of the main functions of this redundancy was to make the DNA code very optimal in terms of resisting bad effects from point mutations. So to make it sort of impervious or less susceptible to both point mutations and frameshift mutations, this redundancy was critical. So for example, if we have CUU, and as we're copying it, that gets accidentally changed to CUC, that's okay because that point mutation will not have a drastic effect because both of them code for the amino acid leucine. And we covered all of this when we talked about the optimality of the genetic code and how it was so optimally designed to resist point mutations and frameshift mutations. But it turns out that at the same time, this redundancy has a very, very important function in regulating the speed of proton synthesis. So for example, CUU, when it is found on the messenger RNA, yes, it will code for leucine, but it will probably take longer or shorter for that leucine to be hooked on than if the codon was UUA. So yes, leucine is hooked on in that position in the protein, but it is hooked on with a different speed. And that is what we mean by controlling the speed of protein synthesis, and that the redundancy of the genetic code also serves that function. Okay, so let's take a look together at this paper. You see that it's published in 2017, and it's called Synonymous Codons, Choose Wisely for Expression. And it is basically, again, getting at the idea that, let's begin reading the abstract together. The genetic code, which defines the amino acid sequence of a protein, also contains information that influences the rate and efficiency of translation. Neither the mechanisms nor functions of codon-mediated regulation were understood. And I'll stop here and I'll leave the rest of the abstract for you, whoever is interested, because then it really starts to get into some nitty-gritty that is beyond the detail that I want to go into. But it is talking again about how just recently we are beginning to discover these mechanisms behind codon-mediated regulation. And again, what does that mean? It means that when I am using synonymous codons, yes, all of them may code for leucine, but the DNA seems to choose very wisely, because a protein that needs to fold more slowly, it will choose a codon that brings in the leucine more slowly, so that that protein comes off the ribosome more slowly. And so yes, leucine is found in a lot of different proteins, but it turns out that the DNA uses the appropriate codon to bring in leucine or any other amino acid into the different proteins based on the rates that those proteins need to be made so that they can be properly folded. And not only that, but remember, a cell doesn't make just one copy of a protein. Your pancreas doesn't make just one insulin molecule. It needs to make a lot of insulin. And how much is influenced by the stability of the mRNA. As I am taping this, we're in the middle of the COVID epidemic, and I'm sure you've heard about the mRNA vaccines from Pfizer and Moderna, and people were saying that the mRNA gets in your cell, it will get translated to a spike protein for the coronavirus, and then it will break down. And so it needs to stay around long enough to make enough spike protein, but then it needs to be degraded so that your cells don't keep making spike protein. So the mRNA needs to be stable at the right level. It needs to last long enough, but not too long. And it turns out that part of the codon-mediated regulation function also affects not only the speed of translation, but the stability of the mRNA that contains that codon. So one synonymous codon may be more or less stable. It makes the mRNA more or less stable than another synonymous codon, even though they both code for the same amino acid. And that concept is what this paper here from 2016 is showing. And it is basically saying that part of what these synonymous codons do, choosing the right codon at the right place in the mRNA sequence, influences mRNA stability. So let us begin reading this abstract together. It says the genetic code was deciphered more than 50 years ago, but we are only now becoming aware of a second hidden code. It is the concept of codon optimality, choosing the right codon at the right place at the right time, even though multiple codons would code for the same amino acid. So it is the concept of codon optimality that enters the scene of developmental and homeostatic gene expression, linking translation rates, mRNA stability, and tRNA abundance. So let us stop here. I think this really says it all. And so we have seen that, when these authors are talking about code within a code, that is different than another code within a code that we've seen, for example, influence the nucleosome structure and the winding of DNA around the spool that makes the chromosomes. And so we will actually see that it's not just a code within a code, it's a code within a code within a code within a code, and every time a new discovery is made, we will see how the media sort of trumpets and says, hey, there's a second code, but there have been multiple second codes. So it is not one layer of embedded information, but multiple layers like an onion. So we've covered a couple. And in the next lectures, we will look at another couple. Of course, we can't cover everything, but we want to bring in enough detail so that we really get a feel that we're understanding what's going on and glimpsing part of the magic.